Today we are going to cover the topic of transcription from ncert. We are going to try and make it much more easier through the points which are given over here. First of all, transcription, which is there, this process indicates the copy of information from DNA, okay, to the RNA. Genetic information which is there is transferred from DNA, from one strand of DNA to RNA. Not both of these strands of DNA are not used, and the reason for this also, there are two reasons. Okay, if we see in DNA replication, both the strands of DNA were used to make another DNA, but to make DNA convert into RNA, only one of its strand is used. The reason for this is basically DNA which is there, if both the strands, this and this strand, okay, one is 3' to 5' and one is 5' to 3'. If this strand which is there makes an RNA, okay, then it would code for a protein, okay, and this would be protein 1. Suppose we name it as protein 1, and another strand of RNA would code for a protein. So there would be two different proteins for the same strand of for the same DNA, okay, which would complicate the genetic process. This is the first reason, okay, this is the first reason why both the strands are not used. The second reason is, first, if RNA is prepared at the same time, the word used in ncert is simultaneously. When RNA is prepared simultaneously, then the thing happens is both the strands are complementary. We know whenever strands are complementary, they make base pairing, as a result of which we cannot separate these strands and they cannot undergo translation. The process after transcription is translation, right? They cannot undergo those processes. As a result, no protein is formed. So as a result of which, this is also not useful where both the strands of DNA can't be used. Now we will see what is a transcription unit. A transcription unit includes three things. First is promoter, second is the structural gene, and third is the terminator. Each part has its individual function. First we look at the promoter. The name indicates that it is the first part, okay. So this yellow part over here indicates the promoter, and the red part over here indicates the terminator. The promoter is the part, okay. Suppose we see that the most important person which was present in DNA replication was DNA dependent DNA polymerase, which indicates that from DNA we make DNA, but from DNA when we are converting it into RNA, then the most important person over here is DNA dependent RNA polymerase. That is, from DNA we are making RNA. So DNA dependent RNA polymerase, we make it as this symbol, okay, so that it may be easier. So this person who is there, he comes towards the promoter region. Always he recognizes a structural gene from its promoter, so it comes towards the promoter region, okay. And in the transcription process itself, there are three processes: initiation, elongation, termination. And these three processes which are there, we are going to see later on in detail. Now the DNA dependent RNA polymerase which we saw, it binds to the promoter region. So this means that promoter is the binding site. What is promoter? The binding site for RNA polymerase. So the first function of promoter is the binding site to RNA polymerase. So it binds over here. The second function of promoter is it defines two strands, okay. It's known to define two strands. Now what are these two strands? They are named over here. First is template and coding strand. DNA dependent DNA polymerase always binds to one of the strand, which is the template strand. So the strand from 3' to 5' is called as template, okay, and 5' to 3' is known as coding. Another function of DNA dependent RNA polymerase is that it codes from 5' to 3' always. मतलब इसे हमेशा 3' to 5' strand से opposite ही रहना पड़ेगा. So it is quite important to notice this function of the polymerase. Unless and until we don't look at this function of the polymerase, we can't define these two strands. only when it goes from 5 prime to 3 prime direction, only then can the 3 prime to 5 prime strand called as template and the 5 prime to 3 prime as coding strand. So we got the two functions of the promoter. The terminator has only one function, that is end of transcription. So it's just responsible to end transcription process, and that is how the promoter and terminator are defined. Now we'll look at the structural chain. What is the structural chain? It is this part between the promoter and terminator. This entire sequence which is present, this is structural chain. In NCERT it is given that promoter and terminator are flanking. What are flanking? They are present at the at the ends, okay, at the opposite ends of the structural chain. Another thing is that terminator location which is there in the NCERT, it is given, it is 3 prime end downstream. What does it indicate? That 3 prime end downstream of the coding strand, means with respect to coding strand it is towards the 3 prime end, and promoter is at 5 prime end upstream. So this is upstream and this is downstream. So it is 5 prime end upstream of the coding strand. So the we can say the address of the promoter and terminator is given with respect to the coding strand, and towards the left it is always upstream, towards the right it is always downstream. So this is all which is given in the initial part. After that there are additional sequences, okay, present before and after the promoter and terminator. So here this region also has certain sequences. Their functions are not so much well defined over here, but they are responsible for efficient process of transcription. Now later on we will see that gene which is there, it is a functional unit of inheritance. Now what is a cistron? Cistron is also known as gene, okay, or it is also known as a polypeptide, a segment of DNA which is coding for a polypeptide. Polypeptide means protein. Many peptides join together to form polypeptide, and peptides together join to form protein. So as a result, when certain part of DNA, okay, suppose this is the DNA and this part of DNA is said to make a protein, okay, so this part of DNA is called as cistron, or this part of DNA is called as gene. That's the basic definition of gene. After that, certain times there are more than one cistrons, and when there are more than one cistrons we are calling it as polycistronic, and if only one, then monocistronic. Polycistronic condition is seen in bacteria and prokaryotes, okay, bacteria and prokaryotes, and monocistronic is seen in humans, or we can say eukaryotes. We are also included here. So in eukaryotes only one functional segment is present which codes for a polypeptide or a protein. After that, they have also said that exons and introns, certain things which are present in eukaryotes. What are exons? Exons are those parts which are said to be functional, which are said to be utilized or are appearing in matured RNA. But introns are not present in matured RNA. This is seen only in eukaryotes and not prokaryotes. Prokaryotic transcription therefore becomes very simple because there are no interfering introns present, as a result of which there are three processes: initiation, elongation, termination taking place back to back. But in eukaryotes there is a hindrance, and how we'll remove this hindrance? See, first of all we'll see the eukaryotic and prokaryotic transcription difference, okay. Prokaryotes, we know they do not have well-defined nucleus. As a result, the process of transcription which takes place in nucleus, okay. Then in transcription, the initiation, elongation and termination process occurs. Okay, in transcription these three processes are included. After transcription, this is first process, there is translation. Okay, first we look transcription. When these three processes occurs, what happens in initiation is this is suppose the segment, okay, of mRNA, And here this is the RNA polymerase about which we described. This RNA polymerase, which is the, suppose this is promoter and this is terminator. It binds to the promoter region. And when the initiation process occurs, okay, when initiation occurs, at that time there is a factor called as sigma factor, which binds with RNA polymerase. Because RNA polymerase cannot differentiate initiation and termination. It only can work during elongation. Okay, this is an important point. It works only during elongation. During initiation, sigma factor comes, and during termination, rho factor accompanies it. Only during elongation process, it's working individually. So as we were looking at the first process, so during initiation, we saw sigma factor binding. So this is initiation. After that, the second process comes, which is elongation. It itself tries to bind to the structural gene. Okay, this is promoter and this is terminator. The last step is termination, where it's here, and over here there is a rho factor, okay, which is binding to it, which indicates there is end of transcription. During this process, RNA polymerase, which is there, it's going along the length, okay, and when it's going along the length, it is transcribing, that is, it is creating RNA from DNA. So this strand of DNA which is there, over which RNA polymerase moves, okay, when it binds to the end termination, at that time this RNA polymerase is released in the nucleus. So this entire process is occurring in the nucleus, okay, transcription process which we saw, and the 3 steps in it. After transcription, the translation process occurs. One factor that we need to remember in prokaryotes is, as the nucleus does not have a well-developed compartment, transcription and translation can be coupled, which indicates both the processes can occur at the same time without the transcription process getting over. And both the process can occur in the nucleus itself or in the cytoplasm. There is no differentiation, nucleus or cytosol. There is no difference where this process occurs. After that, other points which have been mentioned over here are, we have covered them, okay, transcription and translation. So we saw how the prokaryotic RNA functioned. Now we'll look at eukaryotic RNA. In case of eukaryotes, basically, first here also transcription occurs, but we saw there is presence of introns which are interfering. So to remove them also, there are 3 steps which occurs. First is capping, okay. Before capping, we are going to do splicing, then capping, and last is polyadenylation, annihilation. After this process occurs, then we undergo transcription process, and finally translation process. Suppose this is the DNA sequence, okay, present in eukaryotes, and this amount of part which is there, we need to remove. So in splicing, they cut this part, it comes out, okay, this part comes out, and then it's cut off from the rest of the DNA. As a result, the unwanted intron is removed. Similarly, this region which we cut off, now to join these 2 regions, okay, like that, to join these 2 regions which we saw, this and this end, to join them, DNA ligase is used, okay, and it connects the ends. Similarly, entire DNA part where transcription is taking place to form RNA is undergoing splicing to remove the interfering introns. After that, capping occurs. In capping, we'll first mark it as 3' to 5' end. In capping, the 3' end and the 5' end needs to be well defined, because always the 5' end which is there is the place where methyl guanyl phosphate is going to be added. Methyl guanosine which is there, triphosphate, is added at 3' end, and at 5' end which is there, polyadenylation takes place. Here methyl guanosine triphosphate, okay, MGT, and at 5' end we are going to add adenylate residues. About 200 to 300 are said to be added at 3' end. Such sequences are going to be continuously added, and these sequences are added in a template independent manner. This word is important. So this was all the steps involved before transcription. After that, transcription, similar here also 3 steps takes place: initiation, elongation, termination, and finally translation takes place, where this RNA which is obtained is converted into protein, which we would see later on in next upcoming video.